Okay. Looks like a few more people just came in there. All right, well, I think we can go ahead and get started. Yeah, Jody, is everything good? Good to go?
Everything looks good. And hello, everyone. Welcome to today's webinar given by Charlie Rocco from Parse Biosciences, and he will be introducing you about single cell 3.0. Over to you, Charlie.
Great, thanks. And yeah, thanks again to everyone else who has come to the session. Looking forward to talking to you guys more about single cell sequencing here. Real quick, we can jump into it. So, the first thing, just to give you guys a high-level overview of the company, Parse Biosciences. We are a single cell sequencing company that is now working with over 150 different groups worldwide. And so while the majority of our institutions are spread out across the United States and various countries in Europe, we're very excited to be adding many more institutions across Australia and New Zealand from all of you quite soon. So, we are have a mission here of doing scalable single cell sequencing, and also of making single cell sequencing a lot easier and simpler to do. And much like other genomics technologies that have evolved over the years, as you're aware, single cell sequencing has been evolving over the years as well. And so, in the early days, say 2014 to 2016 here, single cell sequencing was really limited to doing technologies where you're putting one cell into one, say, well here, kind of like the SMART-seq technologies, if you're familiar with that, where, the kind of high scale projects at that point was doing hundreds of cells. As you're likely aware, there was kind of the second iteration in single cell sequencing that introduced microwells or droplet-based technologies, where that was a huge step up, right, from kind of the 1.0, where you can now operate on a scale of thousands to tens of thousands of cells. And what we're really excited as a company to introduce to you is really kind of the third generation of single cell with split pool combinatorial barcoding that now enables really anyone to go from tens of thousands of cells up to millions of cells in a very easy and streamlined fashion. Now, we do have a family of kits here to do single cell RNA sequencing. And so this is all under the Evercode line. So we have the WT Mini that can do about 10,000 cells across up to 12 samples. We have the standard WT, which can do 10 to 100,000 cells across up to 48 samples. And we have the WT Mega that can go up to 1 million cells across up to 96 biological samples. We will be going into more details about the difference between these kits, but I do want to give you a bit more of an overview of how our technology works before we jump into that. Now, across these kits, we can really, using these three different kits, we can kind of span the spectrum across what is a pretty large scale. And so we really are able to allow you to do, say, pilot kits with the mini kit all the way up to, say, large high-plex, high-throughput projects in the Mega. Across all these kits, we don't have any instrumentation that's required. And so you can buy these kits and really run them in any biological lab. The core equipment you need is a thermocycling machine and a multi-channel pipette. So something that's kind of, I assume most labs these days already have kind of stocked up. One nice aspect to our kits is that we do have a fixation component up front. And so when you think about doing high-plex experiments, maybe if you're doing, say, 96 samples, you're probably never going to have those 96 samples collected all on that same day. And so with the fixation, it's about a 30-minute workflow that allows you actually to fix and preserve your sample for up to 6 months before proceeding to the downstream barcoding steps. And finally, we've made dramatic improvements to the actual gene detection and sensitivity of the method that really allow you to actually capture much more resolution in your single cell sequencing data. And we'll definitely be diving more into that in some future slides here. So before I jump into how our technology works, I do want to kind of revert back to, say, the 2.0 version and just cover, how does kind of single cell sequencing work in a more conventional manner. And so, the way that conventional single cell RNA-seq has worked is you have a single cell suspension. You co-encapsulate one cell into one compartment along with a set of DNA barcodes. Once you have this co-encapsulation event that occurs, you can actually break the cell open and append these barcodes to the end of the transcripts that are released from the cell within each of these compartments. Therefore, if you were to, say, pool these 4 sets of molecules all together for Illumina sequencing, you can understand the cell of origin of each molecule by a function of that barcode sequence at the end of the transcript. Now, the way that Parse works is a little bit more elegant. And so rather than introducing a new compartment for every new cell, we actually use the cell itself as a container for its own molecules for barcoding. The way we actually do this is by taking the same single cell suspension you saw in the beginning of that previous slide. We then conduct a process called fixation and permeabilization. What that does is it actually anchors the RNA inside the cell, but then we chemically poke holes throughout the cell wall and cell membrane that allow us to flow our own barcodes and enzymes to that cell wall to then act on the RNA. With that in mind, we can then subject cells to what we call split pool combinatorial barcoding. This is a technology where the proof of concept was published here in Science about 3 or 4 years ago. And I can tell you a bit more about how it actually works right here. So what you do is you take the cells that have been fixed and permeabilized that are shown here on the top left. You distribute them across this first barcoding plate, where there's anywhere from 12 to 96 wells available, depending on the kit that you'd be using. And so each of these wells has a well-specific barcode associated to it. But if you zoom into, say, one of the wells here, say the blue well, for example, you'll notice that there's many cells in each of these wells. And all the molecules within these cells are getting that same well-specific barcode appended to those transcripts here. So you can see here, there's many cells in this blue well, and all the RNA molecules, all the RNA transcripts are getting this blue well appended to it. So we're not uniquely barcoding anything quite yet. What we can do is we can actually pull the cells together from that first round of barcoding and distribute them, split them up again into a second round where there's now 96 wells, again, each with its own well-specific barcode. And maybe you can begin to appreciate that cells are starting to take a unique path in this process. So a cell on the top left came from a blue well in the first round, whereas a cell on the right here came from a pink well in the first round. And very importantly, the unique path that cells are starting to take in this process is actually being documented through this combination of barcodes in the cells here. So we can actually conduct this process 4 times. So we have another round of 96 wells in our third round of barcoding. And in the fourth round, we can have up to 16 different tubes here that we call sublibraries. And each of these, what you're going to do actually right before you do this final split here is actually count your cells. And you can distribute, any number of cells anywhere from, say, a few hundred to up to 62,000 cells, depending on the kit that you're using, into these sublibraries. The nice aspect here is that you can actually use these sublibraries to toggle between how many cells you might want to sequence on a specific Illumina sequencing run. After you've done the barcoding here, you will notice that we've generated on the order of up to 14 million total barcodes here. And so if you'll notice that this is an exponential scaling event. So 96 times 96 times 96 times 16 across these 4 rounds of barcoding is how we actually get to that 14 million barcode number. And as a reminder, this is 14 million total paths, essentially, that a cell can take. And so by sequencing far fewer cells in this, say 1 million cells, this is how we're able to get single cell sequencing, despite never actually separating one cell from each other throughout this process. Now, we do, in the kit, provide quite a bit more than just those plates. We actually do provide the reagents to go through the entire workflow from a single cell or nucleus suspension through to an Illumina sequencing library. And we do also provide a computational pipeline to do the sample demultiplexing as well as data visualization and analysis. And I will be jumping more into this later on in the presentation. I do first want to talk about some of the data here. I just told you that we can do single cell sequencing without ever actually isolating a single cell throughout that whole process. So obviously you might want to know, how do we actually determine that we are truly doing single cell sequencing? And what we do here is we subject cells to a pretty simple QC experiment. We call this a barnyard or species mixing experiment, where what we've done is we actually combined a mouse and a human cell line together in one tube. We then conduct the fixation and downstream barcoding and library preparation steps. And we can actually generate a plot here that you see in this slide, where every dot is a perceived cell from the method. And ideally, what you want to see is many blue dots and red dots, as these are either true human cells or true mouse cells, because you're essentially acquiring just one set of species in a perceived cell. Now, if you do see any dots in the middle, this is actually going to be kind of a collision or a multiplet. And so when we do actually this experiment on a Evercode WT kit, this is across 100,000 total cells, we do actually see an observed collision rate of 1.7%. We have done this experiment across the other lines of the Evercode series, both the WT Mini and the WT Mega, and we observe highly similar collision rates here. It is worth pointing out that all these collision rates are about 3 to 4-fold lower than what you expect in a droplet scale method here. Now, one of the things that we really wanted to quantify when we were developing this technology is that we don't conduct any bias or impart any bias on different cell types as we're going through the method. And so what we did here is we took a PBMC sample. We actually get this cryopreserved from a local blood bank where they've actually run flow cytometry before we ever got the sample. We actually do take that sample, we will thaw it, we fix it, we then kind of put that back in the freezer until it was convenient for us to do the entire workflow. And once we do finish the workflow, we're actually able to compare the flow cytometry data that the blood bank collected to what we see after we've done the entire workflow and look at the RNA-based markers here. And so what we do see across the board here, across various different cell types that you'd expect to see in PBMCs, is we have very similar proportions across the board. So it's just, it's something that you'd expect to see, but unfortunately, it's not always the case in other single cell technologies. A kind of another use case here is enabling high-plex time course experiments. This is a project conducted by the Allen Institute for Cell Science, where they were doing a time course experiment with induced pluripotent stem cells, going from the naive stem cell state all the way to a cardiomyocyte. And what they were doing is measuring actually different edits to cell lines in combination with different differentiation protocols to really best understand what was going on in their experiments. And to really make sure they knew what was going on, they actually did replicates for every time point as well. And so across these 4 time points of day 0, day 12, day 24, and day 90, and the other conditions they were doing, they actually had 48 different biological samples. And the nice thing here is that with the fixation, they're actually able to, on each of these time points, just conduct that 30-minute fixation step, throw their sample in the freezer. And after these 3 months went by, all these 90 days went by, they were actually able to pool all 48 samples into a single kit and get all their data back out in one go. So, this is not only a huge time and cost savings. This would have been probably 6 or 7 different droplet-based experiments if they're going to do it that way. But more importantly is it's really more reproducible and kind of higher quality data here, because you can actually reduce batch effects dramatically by having everything in the one run. So instead of having kind of 6 different runs where maybe they're done on different days with different temperatures, and you now have to apply batch effect correction across all of these different data sets, we can now look at the differences between these data here and know with certainty that the differences we're seeing are truly biological, because it was all done in the same barcoding run. Now, one of the valid criticisms that we got when we initially published the proof of concept paper is that our gene detection was just far too low. And this is really something that we've been heavily focused on for the last 3 or 4 years here. And so, what I'm showing here is 2 different plots. It is from the same data set of HEC293 cells. And so in our 2018 paper here, we were getting at full saturation of sequencing about 15,000 unique transcripts per cell. And you can contrast that now to the chemistry with Parse Bio, where we're now getting about 90 to 100,000 transcripts per cell, where we're not even actually fully reaching saturation at this point. This does reflect, as you'd expect, on the genes detected as well, where in our proof of concept, we're getting about 5,000 genes detected per cell. And with the updated chemistry, about 11 to 12,000 genes detected per cell. So about a 4 to 8x increase in transcripts and about a 2x increase in genes detected. This is gene detection that we actually see consistently across all 3 different kits that we do have in the Evercode line here. And so to give you a better sense of this, we're showing you gene downsampling plots, where the x-axis is looking at the raw sequencing reads per cell. So we're just looking at total number of reads from an Illumina sequencer. And the y-axis is looking at how many what's the median unique genes detected per cell. And we see that we have very high correlation here between the mini, the WT mini, the WT, and the WT mega. So as you scale up, you won't have any compromises on data quality. Another question you might ask is, how does this stack up against, say, droplet-based methods here? And when we apply the same data we saw on the previous slide here, but also with different sample types, and compare that to droplet-based competitor, we see that we are actually exceeding them across multiple sample types here, really across the board. So there's PBMCs. This is here on the top right is mouse brain nuclei, NIH3K3 cell lines, and mouse heart nuclei. And you might ask, so what does this really mean? Why is this important? And when it comes down to the sequencing aspect, it can really actually save you quite a bit of sequencing. And so if you just look at the PBMC data set here, for example, let's say at 30,000 reads, you're getting about 1,700 genes per cell with, say, a PBMC data set. But you might ask, well, where do we land to get that same amount of information from a PBMC? And we would be at about 17,000 reads per cell, so almost half the reads per cell required. And what that really means is that you can either do almost half the amount of sequencing to get the same data, or maybe in a more interesting perspective, actually doing twice the number of cells for the same sequencing capacity. I do want to take a step back and just demonstrate, really the power of doing high-scale experiments, and particularly a use case here for the WT mega. And so, for us, what we pursued here after developing the WT mega product is developing a comprehensive PBMC data set for type 1 diabetes. This is a disease that actually affects over 3 million different Americans here. And I actually don't know the number for worldwide here, but it is a pretty profound condition. And there isn't a lot known about it. And partially it's because there's been a lot of data sets that have done, a few different PBMC samples, but not enough to really confidently know exactly what's going on or making differences here. And so what we were able to do is work with our local blood bank. And much like most high-plex experiments, we collected samples on different days, actually over the course of 3 weeks. Our R&D team here collected samples on the days you see here, and actually did that 30-minute fixation step and threw them in the freezer. And after these 3 weeks went by, we then proceeded to do a WT mega kit, where we profiled 12 healthy donors and 12 type 1 diabetes donors, which in aggregate, it came up to just a touch over 1 million total cells here. Doing some initial cell type identification, we do see that we get all the cell types that you'd expect to see within PBMCs. Of course, there's more metadata here that overlaps the different samples as well and different conditions with these samples. Similar to what I showed you earlier, but maybe at a larger scale here, not just looking at one sample, but looking at multiple 24 different PBMC samples, we again see we have very high correlation between flow cytometry when we compare that to the WT mega kit here. So this is comparing flow markers of T and B cells, so CD3 and CD19, across the so that's protein-based flow cytometry against the RNA-seq based Parse kit here. And really, one of the things I want to highlight here is that, it's across 24 different donors, we're able to get a range of 30 to 40,000 cells per sample. And so, while there's many projects where, say, you do just want to pursue one sample and, just atlas the entirety of a single sample, get 1 million cells, it is worth pointing out that the field is moving towards doing more reproducible single cell experiments where it's not just doing many cells for a single sample, but actually doing many samples where you're still getting robust numbers of cells per sample. And so we are really excited here to be enabling kind of the next evolution of single cell experiments, such as going to be more robust and more reproducible data here. Coming back to the general workflow. So as I mentioned earlier, you start with your single cell or nuclei suspension. You pass them through the single cell kind of wet lab kits here, can perform Illumina sequencing, followed by data visualization and analysis. The first step of the wet lab workflow is fixation. And so this fixation step takes about 30 minutes to do. And it is worth pointing out that fixation actually enables quite a bit of flexibility in your experimental kind of planning. And so while you can just do, after you collect the sample, you'll conduct that fixation step, you can actually split that one fixed sample into multiple aliquots. And all 3 of the Evercode whole transcriptome kits are supported by the same fixation solution. And so what that means is that you can save maybe 1 or 2 aliquots to perform that one fixed sample on, say, a WT mega down the road. But if you are really burning to do a really quick experiment and run through that real, get some data out real quickly, you can actually take one of those aliquots and put that on a WT mini right off the bat there. It allows you to actually, separate out, your sample collection from even experimental planning. If you're not even sure exactly how many cells you want to be doing, or if you're not, maybe you're waiting on results from an initial experiment before you decide how you want to proceed with the rest of your samples, for example. It is worth pointing out that once you do fix your samples, we have validated that they can be stored for up to 6 months without having any significant degradation to data quality. So looking at a sample that we fixed and proceeded straight to the workflow without any freezing, and comparing that to that same sample that we then froze for 6 months, we see that we retain about 97% of the median transcripts detected per cell and median genes detected per cell. And while it's not in this presentation here, when we do compare the gene expression correlation is quite high, as you'd expect. Now, once you do have your fixed samples, you can proceed to the barcoding and library preparation. And so first, we'll start with the barcoding section, where the input here is anywhere from 1 to 96 biological samples. This will depend on the kit that you select to run. This does allow you to actually run multiple fixed samples, taken from different days in parallel. And the barcoding section here is about 1 day long here. It's about 6 to 7 hours to perform. Once you've conducted that step, you can proceed to the library preparation workflow, which is a series of fairly standard library preparation steps here that will bring your sub-libraries that we talked about a bit earlier in the presentation into an Illumina sequencing-ready library. And so the end result is a tube of molecules that you can put on any standard Illumina sequencer with a standard configuration, no custom primers. Once you have sequenced your molecules, you can proceed with our computational pipeline. The pipeline starts with FASTQ data and will kind of output 2 primary things. First is an experimental summary report that I'll get into a little bit more detail in the next slide. And the second is cell by gene count matrices. This is kind of or in other words, some people call it a digital gene expression matrix. This is kind of the input to Seurat, ScanPy, and other very useful open source single cell analysis tools that you can definitely do more custom analysis in. We do also provide tutorials for Seurat and ScanPy in particular that help you in a very streamlined way take the outputs from our pipeline and start producing more analyses in these 2 different software packages. The pipeline can be downloaded and run locally on your cluster as well. And we also do have a bioinformatics support staff that can help you in case you have any difficulties setting that up. To give you a sense of what the experimental report looks like, we do have kind of some screenshots here. Now, in reality, it's quite interactive. So you won't be able to see that in this exact data set here, but do encourage you to go online and see the full report yourself. We do print out some pretty standard QC metrics here, indicating how many cells we see per sub-library or per experiment, as well as how many unique transcripts and unique genes are detected on a per cell basis. We recapitulate the saturation curves that we also discussed a bit earlier that you see here on the right. In addition to kind of more QC-based metrics, we do print out preliminary clustering, as well as differential gene expression analysis for those clusters. And so you can kind of browse by cluster name, different samples if you multiplex samples, and you can also type in a specific gene of interest and see where that falls on the cluster map. And so in this particular case, I believe we're looking at an excitatory neuronal marker, and this is a mouse brain nucleotide data set. And so, for folks who'd like to move forward here, this would be in collaboration with Decode, of course. You'd have a standard onboarding with a field application scientist here, where we would walk through the workflow here, go over some key steps in the protocol, and of course, make sure you are fully set up and very comfortable and ready to go into the protocol there. We also provide access to the customer support suite. And so this has additional videos and tips for the wet lab workflow, in addition to pipeline documentation, and that's also where the pipeline actually lives. And so kind of everything else you need to do the downstream analysis section. With that, I'd like to thank you guys all for your time here, and I'm happy to answer any questions you may still have.
Let me see the chat box. So there is a question in chat box. So the first one is there any difference in performance for processing single cell versus nuclei?
Yeah, it's a good question. So the only difference is we have two different fixation kits. One is a nuclei fixation kit, and the second is a cell fixation kit. Both of those are compatible with all the Evercode whole transcriptome kits. And so you'll just want to make sure you order the right fixation kit up front, and everything else will be the same.
And there is another question: with a minimum of around 800 cells at 12 sample, what is the cost per sample? And by comparison, cost for doublet approaches is 50 per sample. Did you get it?
I believe so. I will definitely defer to the rest of the Decode team for that one to make sure we answer the price question appropriately. It will depend a bit on the specific experimental design there, and so we want to make sure we hammer that out before we make sure we have the right pricing there.
And another question is, can it work with fresh frozen blood and tissue samples already collected?
Yeah, definitely. So, that's something we'll definitely want to talk about a bit in the onboarding call, make sure that everything's set up correctly. But actually, the PBMC data that we showed you today is with fresh frozen blood. There's been a number of other groups who have had other tissues kind of already banked that have successfully run the workflow from those previously banked samples. And so if they're all fresh, that's pretty easy. You want to, of course, make sure that's in a single cell or single nuclei suspension before proceeding to the fixation step. If there's kind of other formats, we'd be happy to discuss in more detail to make sure that it'll work appropriately.
And there is another question: how many cells in a sample for splitting you can use?
How many cells? And maybe that, just a clarification, or I'll say I'm assuming that the question is asking like how many cells will need to go into the barcoding workflow. And so this number does vary a bit based on the solution that you're plan on using, either the WT Mini, WT, or WT Mega. And so the retention kind of ranges around 30%. And so the number of fixed cells you plan to put in, you should expect to recover about 30% in your cells recovered.
And are any plans for cell surface protein like site-seq or attack-seq?
Yes, so we definitely see a lot of value in collecting multimodal information from single cells. We did choose to start off with single cell RNA sequencing, as we know it's a very valuable modality, but it's not the only one. And so we do have plans for multimodal measurements in the future, and would encourage you to stay tuned.
And what's the extent of sample loss during the steps of multiple rounds of spread barcoding?
Great, and that may be that's similar to, I believe, the previous question regarding the sample loss here. And so yeah, it's about 30%. So from fixed cells going into cells that you end up sequencing and bioinformatically analyzing, it's about 30%. If there's more clarification desired there, please do elaborate in the chat.
Mm-hmm. Yeah, great. And there is one more question: can it be adapted to pick up, I believe this is gRNA's guide RNA?
Yeah, it's a great question. And so that will, the short answer is yes, it will require some changes to the existing workflow. And so that would be probably a discussion you want to have offline here about how you go about doing that.
I guess that's it for now. There are no more questions. Thank you, Charlie, for a great talk, and thank you all for your time today. Thank you so much.
Great. Yeah, thank you. And do really appreciate everyone coming out. If there are any more questions here, please don't hesitate to reach out, via email to the Decode team or the Parse team here. Looks like actually there's one more question here. Kwan, you might need to elaborate. It says, is there a variation in linking different barcodes? I don't know if I quite understand the question, but I think,
I don't know. Can I just?
Yeah, go ahead.
So like that, like 4 rounds of barcoding, and in each layer multiple barcode sequence. So like in the past when we do like PCR sequencing, usually we see the variation using different barcode sequence. Like some sequence have preferential linking. So do you observe something like that in this protocol?
Yeah, it's a good question. So we have heavily optimized the barcodes here to have very minimal variation. That's a very good question, Juan, and that does happen. But basically, we've done a few years of optimization on the barcodes to make sure that this is extremely minimal and fairly negligible, actually, in the grand scheme of the data.
That's great. Thank you.
Yeah. Oh, and I believe there's one more question.
Yeah.
Go ahead, Jody. Yeah.
So the question is, when will 1 million PBMC data be released?
Yeah. And I apologize, I don't think I actually mentioned it in the talk, but we do plan to release that by the end of the month here. And so that will be available online here, similar to the existing data sets we do have on there for the WT workflow. So we encourage you to stay tuned. They'll be up very soon.
I think we have no more questions. I think that's it for now. And thank you all. And please feel free to reach out to us if you have any other questions. And thank you, Charlie, once again for the great talk. And
Thanks, everyone. Have a good rest of your day there.